Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right. So we're in the process of discussing IP3 receptor signaling, okay, and we've discussed this complicated gating mechanism for the IP3 receptor in which IP3 binding to these IP3 binding sites on each of the four subunits of the IP3 receptor tetramer causes a change in conformation in each of those four subunits, which changes the affinity of two other binding sites, which are both for calcium. Okay, now you have an inhibitory calcium binding site and a stimulatory calcium binding site. So the inhibitory one's in blue and the stimulatory one's there in red. Okay, and when calcium binds to the inhibitory binding site, that keeps the channel closed, and if you can get calcium just bound to the stimulatory binding sites and not to the inhibitory binding sites, then that causes it to open. If you get it bound to all of them, so both the stimulatory and the inhibitory ones, then the inhibitory ones override the stimulatory ones, okay, and that causes the channel to close. Now, prior to IP3 binding then to the IP3 receptor subunits, what is the case is that the inhibitory calcium binding site has a high affinity for calcium, and the stimulatory calcium binding site has a low affinity for calcium. So that means that normal resting calcium concentration within the cytoplasm of 100 nanomolar, you end up with calcium bound to the inhibitory one, which is the high affinity one, and not bound to the stimulatory one. So that causes the IP3 receptor to be held in a closed state, basically. Okay, when IP3 arrives, when you've generated IP3, four IP3 molecules can then bind to these four IP3 binding sites on the four different IP3 receptor subunits. Okay, that causes a conformational change in each of the four subunits, which then leads to the inhibitory binding site losing its affinity for calcium, so becoming a much lower affinity binding site for calcium, and the stimulatory calcium binding site gaining affinity for calcium, so becoming a much higher affinity calcium binding site. Okay, so now what has to happen is four calciums from the cytoplasm have to bind to these four calcium binding sites, the stimulatory ones, that is. Okay, now that's not easy. Okay, that takes time because 100 nanomolar is quite a low calcium concentration, and these stimulatory binding sites, although they now have a much increased affinity, it's still not that great for calcium that it will instantly bind calcium at a free calcium concentration of 100 nanomolar. Okay, however, we've discussed the fact that this leads to propagation of calcium signals, because if you've got a bunch of IP3 receptors with IP3 bound to them, which are these ones in orange here, then what you now have to do is wait for one of them to just happen to bind four calcium arms to its four stimulatory calcium binding sites. Okay, and I've discussed in the previous video about how we don't actually know whether it is four IP3s, four calcium stimulatory binding sites which need to be occupied in order to open it, but we're going to assume that that is the case to simplify matters. Okay. Right, so we are assuming that all of these have got IP3 bound to them, and then we're waiting for one of them to get four calciums binding to its stimulatory binding sites at the resting cytosolic calcium concentration. When it gets that happening, and we're saying it's the one in the middle first, then it will then open and it will release calcium from the lumen of the endoplasmic reticulum into the cytoplasm, and now that calcium can spill over onto its neighbours, occupy their stimulatory binding sites, which will cause them to open, and then it can propagate onwards like so until you reach the point where you get to IP3 receptors which don't have IP3 bound, and then of course when the calcium spills over to those, it will actually potentiate the closed state rather than causing them to open. Okay, right. In addition, we've discussed how when calcium builds up to a high enough level around each one of these IP3 receptors, it will then cause the IP3 receptor to close, because when you get to high enough levels of calcium, then you'll end up getting calcium binding to those inhibitory calcium binding sites, which have a low affinity, but still, if you raise the calcium high enough, you'll force calcium into those sites. Okay, so then when calcium binds in those sites, even though you've got calcium still bound in the stimulatory binding sites, the inhibitory binding site overrules the stimulatory binding site and therefore causes the IP3 receptor to close. Okay, right. So we've now discussed local calcium signals called calcium puffs. What I now want to discuss is a type of global calcium signal which is called a calcium wave. Okay, and we'll see how these gating properties of the IP3 receptor can give rise to these calcium waves which propagate across the cell. Okay, right. So to get a cell to undergo a calcium wave then, you need to put it in a high concentration of your agonist. So we were imagining using acetylcholine and the M3 receptor as our example, so we'll stick with that. So let's now have a very high concentration of acetylcholine. So to get calcium waves, we're going to have a high concentration of acetylcholine, whereas to get the calcium puffs, we need a low concentration of acetylcholine. Okay, right. I'm also going to go to representing my cell in a different way, okay, just to make the picture easier. So now I'm going to represent my cell nice and large as a rectangle here, okay, like so. And then I'll put the endoplasmic reticulum here, okay, stretching the length of the cell basically. Okay, this is just to simplify down the picture. Okay, we could do it on the more complicated picture that I had previously, but this will simplify it down, and calcium waves, they need simplifying down because they're not the easiest things in the world to understand. Okay, right. So the idea here then is that when you have a high concentration of the acetylcholine, then the idea is that loads of the M3 receptors are going to be activated. Okay, nearly all of them. So if I now represent the M3 receptors once again by purple dots on the plasma membrane here, and you get the idea, they'll be all over the cell. Okay, a lot of them are now going to get acetylcholine molecules bound to them. A few might still not have the acetylcholine, but loads of them are going to get acetylcholine bound to them. Okay, that means then that you're now going to get alpha Q/11 subunits being activated all over the place. Okay, you're going to be getting phospholipase C beta enzymes activated all over the place. You're then going to be getting PI45P2 being cleaved down to diacylglycerol IP3 all over the place. So you now get what's called a global IP3 signal basically. Oops, missed off the L. Global IP3. Okay. So IP3 basically goes up all over the place within the cell. Okay, so it's going up everywhere. Now what does that mean? That means basically that most of the IP3 receptors in the endoplasmic reticulum are now going to have IP3 bound to them. So let me now draw this here. So here are our IP3 receptors, and they're all going to have, let's say, 4 IP3 molecules bound to their 4 IP3 binding sites. Okay, and that will mean that these IP3 receptors are now all primed to open if they end up getting 4 calciums binding to their 4 stimulatory calcium binding sites. Okay, so I'll represent all of these IP3 receptors which have the IP3 bound to them once again in orange here. Okay, so now you can probably understand how we're going to get calcium waves now, okay? Because all we need to wait for now is one of these IP3 receptors just to happen to get 4 calciums from the cytoplasm binding to it basically. So we just now have to wait for 4 calciums somewhere to bind to one of these IP3 receptors. And let's just say, just to have an interesting calcium wave, that it's this one over here that happens to get 4 calcium ions binding to it first. Okay, so now of course what it's going to do is it's going to open, Okay, it will then allow calcium to leave the endoplasmic reticulum and go into the cytoplasm, and then the calcium will spill over onto its neighbour. Okay, its neighbour will now end up getting 4 calciums binding to its stimulatory calcium binding sites. It will open and release calcium, and this will continue on, so its calcium will spill over onto its neighbour, and this propagation will continue basically. So you'll get the propagation of the activation of these IP3 receptors right from one side of the cell to the other side of the cell, simply because a lot of the IP3 receptors have 4 IP3s bound to them, and therefore are going to respond in a activatory way to increased cytoplasmic calcium. Okay, now what you're going to actually find is that calcium signals are also going to propagate. So it's not just the opening of the IP3 receptors that propagates, the level actually spikes at different points, and this spike propagates. So let me explain in more detail what I mean by that. Okay, so it's probably best to draw graphs for this. So let's call this here position number 1. We'll call this position number 2. We'll call this position number 3. And we'll call this position number 4, and I won't bother going any further. This will illustrate the point. Okay, right, so now let's draw the calcium level in the cytoplasm as a graph against time, basically. So I'm going to put time on the x-axis here. Okay, and I'm going to put calcium concentration in the cytoplasm on the y-axis here. And first what I'm going to do is I'm going to show this graph for position 1 here, and I'll colour code it up. Right, okay, so what's going to happen is then when this initial event happens, and we'll start the timer from when the initial event happens of this IP3 receptor here opening. So what's going to happen is calcium is going to rise up basically in the vicinity of IP3 receptor at position number 1 here. Okay, so the calcium level in position 1 here is going to go up, up, up from 100 nanomolar down here to some higher level. Okay, and then what will start to happen is as you get up higher and higher, that will trigger the IP3 receptors here to close basically. Okay, so we know that once calcium in the local vicinity of IP3 receptors gets too high, it triggers them to close. So the IP3 receptors in position 1 will close, okay, because they now have 4 calciums bound in their inhibitory calcium binding sites. Okay, and then what will start to happen is the circa pumps in this region, okay, which I'll represent here as this turquoise blob, okay, they will start to return the calcium into the endoplasmic reticulum, okay, and therefore calcium will fall back down gradually in that area, back down to 100 nanomolar, okay, and then of course when you get back down to 100 nanomolar, the calcium will eventually cleave off the inhibitory calcium binding sites, as well as most likely the stimulatory calcium binding sites, and now what we've got here is an IP3 receptor which still has 4 IP3 molecules bound to it, but which is in the closed state because it doesn't have any calcium bound to its inhibitory binding sites, but neither does it have any calcium bound to its stimulatory binding sites. So it is now waiting for some more calcium to bind to it so that it can be opened again, basically. Okay, so that is this spike for position 1 here. Okay, now let's do the same thing for position 2. So when the calcium went up in position 1 here, some of the calcium spilled over to position 2 here and caused those IP3 receptors in position 2 to open. Okay, and that caused calcium to go up in position 2. Okay, but there will be a slight delay from position 1. So here is the rise in position 2, and I'll colour code position 2 in green here. Now, the exact same thing that happened in position 1 is going to occur in position 2, basically. Okay, calcium will go up and up and up in position 2, okay, but then it will get so high that it will then start inhibiting the IP3 receptors in this area. It will start binding to those inhibitory calcium binding sites and overriding the calcium bound to the stimulatory binding sites, and therefore the IP3 receptors in this region will start to close. Meanwhile, the circa pumps in this region, and I'll put the circa pump here, okay, will then start to move the calcium back into the endoplasmic reticulum, and therefore calcium will fall at position 2. Okay, like so. Okay, so you get a spike at position 2. Okay. Then the same thing is going to happen at position 3. So when the calcium rises in position 2, it spills over into position 3, activates the IP3 receptors, which have IP3 bound to them in position 3, and therefore you end up getting another one of these spikes that is delayed of position 2, like so. Okay, so the exact same story holds true in position 3 here, and I'll represent position 3 in blue. Okay, and then position 4, just to add one final one onto here, that will be then delayed of position 3, basically. Its calcium spike will be after the calcium spike of position 3 here. Okay, and we'll have that in turquoise. So basically what is happening here is that you're getting this propagating spike in the level of calcium. Calcium goes up first here, then it propagates onto here, it causes calcium to go up here, it causes calcium to go up here, it causes calcium to spike here, and each one of these areas of the cell undergoing this calcium spike causes the neighbouring area of the cell to undergo a calcium spike. Okay, so this is an example of the principle of saltatory conduction of a signal. Okay, now saltatory conduction is usually applied to action potentials, basically. It's usually the way we describe action potentials, but saltatory conduction is more general than action potentials in neurons. Okay, saltatory conduction is really just the idea that you can propagate a signal by something here causing something here, which causes something here, the domino effect, basically. If you want to send a signal, you can effectively have loads of dominoes lined up, okay, and then you can push it at one end, and then it will propagate on and on and on, and it can send a signal from where you are to where someone else is. Okay, that is the principle of saltatory conduction, that you can send a signal from one place to another place by a disturbance in one little local area causing the same disturbance in the other area, which then propagates on and on and on. So saltatory conduction, really just think of the domino effect, basically. Again, action potentials, normal action potentials in neurons are an example of saltatory conduction, and these calcium waves across cells are also an example of saltatory conduction. And in fact, these calcium waves, some people actually call them calcium action potentials, and there is a lot of interest now in the fact that glial cells, okay, particularly astrocytes in the brain, they do not have normal electrical action potentials, but they do have these calcium waves which can propagate across them, and these calcium waves are capable of spreading from one astrocyte to the next astrocyte, and there is huge interest as to whether these networks of astrocytes are also forming networks, just like neurons form networks, that are capable of thought, basically. Okay, so potentially we've been, thinking that neurons were the cells in the brain responsible for thinking, okay, responsible for computation and thought. But potentially astrocytes are also involved because they have these calcium waves which are very similar to action potentials. They are just this saltatory conducting signal which can conduct onto neighbouring astrocytes, okay, and there is the potential that we have astrocyte networks which are capable of logic gates, basically, just like we have neurons that are capable of logic gates. Okay, so there's a lot of interest in this, basically. Okay, now we need to discuss another thing, which is that it's not normal that a cell will just fire one calcium wave. Usually what happens is the cell fires calcium wave after calcium wave after calcium wave. Okay, so calcium waves in isolation are not normal. Usually you get multiple calcium waves after one another. Okay, so let's talk about how that occurs. Well, basically, once a calcium wave has occurred, okay, and it's passed now, Okay, so this is our calcium wave here. It's passed now. What then can then happen again is, providing the acetylcholine is still present in the extracellular fluid, okay, so I'm assuming we're just leaving that acetylcholine there, then we'll still have IP3, so IP3 will still be high within the cytoplasm of the cell. Okay, and now the IP3 receptors have still got all of their IP3 bound to them. Okay, and effectively we're back to stage 1 now. All you have to wait for is some other IP3 receptor now to just happen to get 4 calciums binding to its stimulatory calcium binding sites under normal cytosolic calcium concentration. Okay, so you now just have to wait a little while, okay, the latency period between neighbouring calcium waves, and then after a while another one of these IP3 receptors, not necessarily the same IP3 receptor that fired this first one, will happen to get 4 calciums binding to its stimulatory calcium binding sites, and then it will initiate the entire calcium wave that will propagate across the cell, basically. Okay, now another key principle then that I want to say here, which is that the concentration of acetylcholine determines the frequency of the calcium wave. Okay, so this determines the frequency of the calcium waves. Now, what do I mean by that? Okay, so basically the frequency means how commonly calcium waves occur, basically. Okay, so it's related to how long you have to wait between one calcium wave and the initiation of the next. Okay, if you have to wait for ages, then that's a low frequency. If you have to wait hardly any time at all, then that's a high frequency. Okay, the calcium waves are occurring very frequently. Okay, now, the concentration of acetylcholine in the extracellular fluid is going to affect how frequently the calcium waves are firing across the cell, basically. Now, why is that? Well, the higher the concentration of acetylcholine, the more acetylcholine molecules you're going to have bound to M3 receptors. Okay, so the greater will be the number of M3 receptors in the cell, or rather on the cell's membrane, that will have acetylcholine bound to it. Okay, now what that means is that you're going to generate more IP3 within the cell. So basically, quite simply, a higher acetylcholine concentration is going to cause a higher concentration of IP3 within the cell. So why does having a higher concentration of IP3 within the cell increase the frequency of the calcium waves? Okay, well, the reason is believed to be, okay, that if you have more IP3, then a greater number of the IP3 receptors are going to have 4 IP3s bound to them. So in this picture that I've drawn here, I have implied that all of the IP3 receptors have got IP3 bound to them. Okay, I've got 4 IP3s bound to them, and are therefore in this prime state, ready to open if only they get 4 calciums bound to them. In reality, that's not the case. Okay, there will also be many IP3 receptors in the ER membrane that haven't got 4 IP3s bound to them, and therefore aren't capable of participating in this calcium wave. Okay, if we have a higher IP3 concentration, then more of the IP3 receptors in the ER membrane will have IP3 bound to them. Okay, and why does that mean that the frequency of the calcium waves is going to increase? Well, if the frequency increases, that means that the time you have to wait for another calcium wave to initiate after the termination of the final one is going to be shorter. Okay, now what determines the amount of time you have to wait for another calcium wave to occur? Well, that is how long it takes for a IP3 receptor somewhere to end up getting 4 calciums binding to its stimulatory binding sites at the normal resting calcium concentration. Okay, if you have now got more IP3 receptors that have got 4 IP3 bound to them, then you have a bigger population of IP3 receptors that are waiting for 4 calciums to bind to them to initiate the calcium wave. So effectively, the chance that one of them is now going to get 4 calciums bound to it is going to be greater. Okay, so what I'm really saying here is, if you imagine having more IP3 receptors with 4 IP3 molecules bound to them, and I'll just put them here now. So I'm adding in now more IP3 receptors that have 4 IP3 molecules bound to them because we now have a higher IP3 concentration. So some of the IP3 receptors that didn't previously have 4 IP3s bound to them are now capable of participating in this calcium wave. Okay, now when you've got more of these IP3 receptors with 4 IP3s bound to them, the chance, or rather how long you will have to wait for one of them to end up getting 4 calciums binding to it, even at the resting calcium concentration of 100 nanomolar, is going to be smaller basically, simply because you have more of these IP3 receptors with 4 IP3s bound to them. Okay, so that's why the latency time between calcium waves might go down, because if you've got a greater number of IP3 receptors which are actually waiting for 4 calcium to bind to them, the probability that one of them will just happen to get 4 calciums binding to it is larger, so it will happen quicker basically. Okay, and that's what triggers the beginning of the calcium wave. You just need one of them to end up getting 4 calciums binding to it, and then the calcium wave will initiate. So basically, the higher the acetylcholine concentration in the extracellular fluid, the higher the IP3 concentration in the cytoplasm, the more IP3 receptors there are going to be that are participating in the calcium wave, basically. Okay, and therefore the chance that one of them is going to get 4 calciums binding to it is greater, okay, and it will occur quicker. So the time that you have to wait for a calcium wave to be initiated is therefore going to be smaller, and therefore you're going to get calcium waves occurring at a higher frequency. Okay, so that then is the topic of calcium waves covered. Okay, calcium waves are very different to that other type of calcium signal that we studied, which was calcium puffs. These are a global signal, whereas the calcium puffs were a local signal. Okay, what we will move on to in the next video is discussing targets for these calcium signals. So we'll discuss CAM kinase enzymes specifically. We're going to look in detail at CAM kinase 2, because CAM kinase 2 can respond differently to different frequencies of calcium signals, basically. So it can respond differently to different frequency calcium waves, which is why it's so interesting. Okay, so we'll talk about CAM kinase 2 and how it can distinguish between different frequencies of calcium waves, and then we'll talk about protein kinase C, and then we'll move on to how the mitochondria responds to calcium going up in the cytoplasm.